Hello, welcome to AP Biology Semester 2. We're talking about big idea number 2, free energy. And today we are in chapter 6, section 3, talking about the eukaryotic cells, genetic instructions that are housed in the nucleus and carried out by the ribosomes. On the first stop of our detailed tour of the cell, let's look at two cellular components involved in the genetic control of the cell. The nucleus, which houses most of the cell's DNA, and the ribosomes, which use information from the DNA to make proteins. Okay, we all know that the nucleus is the information central for the cell. The nucleus contains most of the genes in the eukaryotic cell. Some genes are located in mitochondria and chloroplasts. It is generally the most conspicuous organelle in a eukaryotic cell, averaging about 5 nanometers in diameter. The nuclear envelope encloses the nucleus, separating its contents from the cytoplasm. The nuclear envelope is a double membrane. The two membranes, each a lipid bilayer with associated proteins, are separated by a space of 20 to 40 nanometers. The envelope is perforated by pore structures that are about 100 nanometers in diameter. At the lip of each pore, the inner and outer membranes of the nuclear envelope are continuous. An intricate protein structure called a pore complex lines each pore and plays an important role in the cell by regulating the entry and exit of most proteins and RNAs, as well as large complexes of macromolecules. Except at the pores, the nuclear side of the envelope is lined by the nuclear lamina, a network array of protein filaments that maintains the shape of the nucleus by mechanically supporting the nuclear envelope. There is also much evidence for a nuclear matrix, a framework of fibers extending throughout the nuclear interior. Okay. Within the nucleus, the DNA is organized into discrete units called chromosomes, structures that carry the genetic information. Each chromosome is made up of a material called chromatin, a complex of proteins and DNA. Stained chromatin usually appears as a diffuse mass through both light microscopes and electron microscopes. As a cell prepares to divide, however, the thin chromatin fibers coil up. They condense. In other words, they condense, becoming thick enough to be distinguished as the familiar separate structures we know as chromosomes. Each eukaryotic species has a characteristic number of chromosomes. A typical human cell, for example, has 46 chromosomes in its nucleus. The exceptions are the sex cells, eggs, and sperm, which have only 23 chromosomes in humans. A fruit fly cell has 8 chromosomes in most cells and 4 in the sex cells. All right, I'm sure you've gotten all of these definitions here, so I'm going to move the board so that you can see these definitions here. All right. So, now we're going on to talking about the nucleolus. The nucleolus. A prominent structure within the non-dividing nucleus is the nucleolus. The plural name for nucleolus is nucleoli, which appears through the electron microscope as a mass of densely stained granules and fibers adjoining part of the chromatin. Here, a type of RNA called ribosomal RNA is synthesized from instructions in the DNA. Also, in the nucleolus, proteins imported from the cytoplasm are assembled with RNA into large and small ribosomal subunits. These subunits then exit the nucleus through the nuclear pores to the cytoplasm, where a large and a small subunit can assemble into a ribosome. Sometimes there are 2 or more nucleoli. The number depends on the species and the stage in the cell's reproductive cycle. Recent studies suggest that the nucleolus also functions in regulation of some cellular processes, such as cell division. Excuse me. All right. Now that I've sneezed, we're going to move on to ribosomes. Ribosomes, which are complexes made of ribosomal RNA and protein, are the cellular components that carry out protein synthesis. Cells that have high rates of protein synthesis have particularly large numbers of ribosomes. For example, a human pancreas cell has a few million ribosomes. Not surprisingly, cells active in protein synthesis also have prominent nucleoli. Ribosomes build proteins in 2 cytoplasmic locales. At any given time, free ribosomes are suspended in the cytosol, while bound ribosomes are attached to the outside of the endoplasmic reticulum or nuclear envelope. Bound and free ribosomes are structurally identical, and ribosomes can alternate between the 2 roles. Most of the proteins made on free ribosomes function within the cytosol. Examples are enzymes that catalyze the first steps of sugar breakdown. Bound ribosomes generally make proteins that are destined for insertion into membranes, for packaging within certain organelles such as lysosomes, or for export from the cell, such as secretion. Cells that specialize in protein secretion, for instance, the cells of the pancreas that secrete digestive enzymes, frequently have a high proportion of bound ribosomes. You will learn more about ribosome structure and function in chapter 17. And I'm going to stop here for today. And until next time, ta-ta!